down, guys. Look, thanks for coming back after supper or dinner or whatever it's called in Oxford. I'm very glad you've come back. Tonight is a sort of rejoicing evening. What we do is present two of our medals, and also I'm going to show you a short trailer of our new BSDB film, which the full version will be out in a few weeks, stroke maybe a month. One of the stars in it is in this room, and I'm going to get to stand up later, Courtney. But I'm going to start off with a medal, and so the medal is a new one. In 2019, on the front cover of our BSDB brochure, we had our 3 medals, and those 3 medals we'd had for a while. We still have them, and we're going to hand them out. We handed out the tickle yesterday, you remember, but we felt that we needed one for sort of scientific communication, for education, for outreach, preferably for all of the above. We'd been mulling that over for a little while, and then sadly, you know, one of our grandfathers in the field passed away, and it was obvious that the medal should be named the Lewis Wolpert Medal, because he was an outstanding communicator. We didn't have a design, so we commissioned a design from a C. elegans researcher who's also an artist, called Steph Nowatarski, and our brief to her was, Lewis liked the idea of morphogen gradients, and he worked on lots of model organisms, so could you put those into the medal? And this is the medal, and it's bloody lovely. It probably is the most beautiful of all of our medals. The winner is in this picture from Oxford Developmental Biology unit nearly 40 years ago. And the winner is this guy, who actually doesn't look a great deal older than 30 odd years ago. He's slightly grey-haired. It's Jonathan Slack. And several of his nominators are here as well. So I nominated him, and that's me. So I look quite a lot older. I was a PhD student at the time, and Phil Ingham, who looks about 12 there, seconded. Jeremy Green's not in the picture, but he was the other nominator. And what was clear at the time, I was just a PhD student at the time, but it was already clear to me and everyone in the field that Jonathan's just amazingly clear at you know, getting across a complicated problem and making it, you know, easy to understand. And I bought this book. I was clearly late to the party because I have the second edition. He'd already sort of distilled what was good about developmental biology, and so I learned how each of the different model organisms, what they brought to the table by reading this book. And then he had this novel, which some of you may be aware of, and those of you who haven't read it should. It's slightly controversial, a little bit naughty. It isn't really a novel, it's real life. It's really good. It's hard to get hold of, a hard copy of it, but you can buy it on Kindle now. And I would say it's well worth the read. If you want to understand lab life, particularly of that era, then read this book. So Jonathan hasn't stopped writing. He keeps writing. There's a whole, you know, bunch of developmental biology and stem cell textbooks, but my favorites are these guys. So they're like the sort of mini, what they called, idiots, no, a very short introduction to a topic. And I have stem cells, and I have genes. Genes, you know, it's a huge topic, but what Jonathan is good at doing, as I said, is distilling a big thing and making the key nuggets easy to grasp. And our first recipient of the Wolpert Prize was Jamie Davis, and he only had published one of these. And Jonathan, he's about to write the third, Regenerative Biology. Jamie gave me a copy free of his, so I'm sort of expecting a signed copy of Regenerative Biology when you're ready with it. So So, look, Jonathan, come up and get your medal. Congratulations. You should get him to show it to you this evening because it is a total beauty. Now, Wolpert Medal winners don't give a talk. Rather, they give an Oscar recipients, thank you very much. We don't want any tears. We don't want any politics. You can have a brief time to thank me and others for it. His prize is that he does a lecture tour that the BSDB pays for. So if you fancy a bit of Slack action in the next year, looks like Dundee would like one. So you speak to me and or Jonathan and or Jens, who is our medals man. Okay. See what you think. Listen to what he says.
Okay, well, I've been warned repeatedly that I've only got a couple of minutes. So I thought I'd just show you the rest of the covers of my books on the on here. Now, the thing Paul doesn't know about Egg to Embryo was that the very first copy of the very first edition arrived in my pigeonhole on the same day as the birth of our first daughter. So these were two babies in one day. And of course, guess whose baby had the most attention? Not mine. And Egg and Ego, which is only partly a novel, Paul, not entirely. It has real truth in it as well. I won't tell you anything about it, except I had the original idea while I was on holiday, and I read a book called A Year in Provence by Peter Mayle. And I don't know if the people from Paris are still watching, but if so, this is a very funny book about somebody who does up a farmhouse in Provence in the south of France. And I read this and I thought it was very funny. And then I thought, well, if this guy can write a bestseller about something as dull as building a house, then surely I can write a funny book about something interesting like developmental biology. So that was where that came from. Then there's the textbook, and this has been through 4 editions now, and the current edition was written along with Les Dale from University College, who's a real, he was head of teaching for anatomy and development for some years and brought a great deal to the 4th edition. But this was written first in late 90s when I felt the modern molecular genetic developmental biology had reached a sort of maturity and a consensus about what you should put in a textbook. And I wanted to write something short and sharp and clear, like Egg to Embryo, but at undergraduate level and covering the whole of animal development and not just early development. So that was where it started. But, like everything else, it's grown over the years, partly from publishers' pressure and partly because there's just so much more stuff. So it is a bit bigger than I'd like now. But the key features remain, which is keep the model organisms separate, and then you avoid muddle. Provide evidence for the important conclusions, so it's not just a lot of dogma. And try and keep the amount of molecular detail as small as possible. Harder said than done, I might say. But anyway, that's the textbook. Then I told Jeremy Green I'd mention him, and this was his idea, actually. He noticed a few years ago that not hundreds of thousands, but certainly many thousands of workers in stem cell labs around the world, of whom there are at least 10 times as many as there are developmental biologists, they often know nothing at all about development. And this is ironic because the factors they put on their ES cells were all discovered by us. So he thought there ought to be a book for them about developmental biology. And this is the book. And it also includes a bit of cell biology, sort of basic cell biology, and a few other topics like transplantation immunity. but if you're working in a stem cell lab, that's for you. And then finally, Paul mentioned these. These are very cheap. They're so cheap, Paul, that you can even afford to buy one yourself. And they asked me to do stem cells and genes, and they've both been through 2 editions now. And these are written for the sort of educated layperson. And I must say, I found this very rewarding doing these, because to write for a lay audience, you've really got to understand what you're talking about, be highly selective, and understand the blocks to their understanding. You know, why people can't understand something is just as important as sort of setting it all out for them. So anyway, that's them. And that's the end of the slides. And so I'll just finish by saying thank you again to the committee for this very touching honour. And thank you to my family, who I ignored for many weekends while I was writing all these books. And if there are any of them here, thank you to my former lab members for me ignoring them during the week while I was writing my books. So thank you very much, everybody.
Okay, actually, I could afford one of your books, but I want you to give me one, John, and I want it signed. So now I want to show you this movie, and so this has been on the BSDB committee's agenda for a while, well over a year. We've fancied making a movie. Its purpose is to just flag up and highlight the beauty of the breadth of UK developmental biology, but also to flag up how much of it offers insights that have, you know, clinical translatability, because that's what funders care about and like, and it's what entertains others. The movie is made for you guys to use for outreach. It's 25 minutes long, or will be the final cut. You can chop it up if you want, use it for outreach, or show it to your second years who you're trying to tempt to come and do you know, join your third year developmental biology program. And it features Alice Roberts, who did the introduction for us, and she's sort of brought a bit of star quality and is offering to promote it for us bigger. So she thinks, you know, the BBC might be interested in this sort of stuff. So I don't feature in it, but some of the folk who feature in it might end up big and famous one day. I need to thank folk for their contributions to it. So we've had a working party which included Jeremy Green and I, and we don't feature in it, he and I. Shankar does feature in it, though you're not going to see him in this clip. Rod Young features in it. You're not going to see him. Catherine Brown, she's the executive editor of development. So we're the working group, and we commissioned these guys, Cambridge Filmworks, to do the filming. So it's slick and broadcast quality, and the production, but the words are as, essentially. Courtney Lancaster, who will stand up now, please, is... And you can sit down now. You can sit down now. We selected her to be, she's a PhD student at the LMCB, and we wanted a young roving reporter. So she tours around the UK interviewing various folk in various labs. We've got 5 vignettes. You're just going to see the first one, which is morphogenesis, but there's a bit of plant action. There is neural crest leading into cancer. There's Shankers in the Heart movie or vignette, and there's eye through to the clinic, you know, eye and curing blindness. So it's really good, I think. I hope you like it. I'm just going to set it up so that you guys at the back, can you... Right, I'm going to get out of here. Sorry. what we're going to show you is the first 5 minutes, which is the intro and the first vignette, and we've got a few bloopers which we thought would be fun to show you, and they're at the very end. So we were toying whether we should show the bloopers or not, and Sally said we must show the bloopers. So you get the bloopers. Can I play it now?
The development of a fertilized egg, a single cell, into a whole human being seems to defy belief. It's like some kind of biological miracle. But that egg contains all the information needed to make a baby that will grow into a child and on into an adult. Over the years, scientists have uncovered many of the secrets of that incredible transformation of a single cell into a whole living organism. Cells divide countless times to make tissues that grow and fuse, fold and dissolve in an intricate but reliable way to make a myriad of different living organisms. But development isn't just about cells multiplying. They also need to change into different cell types. This process of differentiation produces skin cells, bone cells, the cells in your liver and your spleen, the cells which form the crystal clear lens in your eye, and the neurons in your brain. In organisms that reproduce sexually, information comes from the joining of male and female gametes. Whether the organism is a plant, an insect, a human, or a zebrafish, development of the fertilized egg produces a miniature version of something about midway between its mother and its father. Understanding this incredible process of growth and patterning isn't just fascinating in its own right, it is hugely relevant to human health and modern medicine. So join us on a journey of discovery into the fascinating world of developmental biology.
So at its core, developmental biology investigates how genes, cells, and the signals they use to talk to each other generate an organism's heterogeneous size, shape, and structural features. This all happens in the astounding but highly reliable process of development from embryo to adult. It tells us about the differences between species and the similarities between individuals, or even twins. Some 200 years ago, Charles Darwin wrote about how embryonic development may be the key to understanding the similarity and divergence between species. From that simple statement, we have gone on to understand that developmental biology involves a complex set of processes, including cell division and migration, patterning and morphogenesis, and cell differentiation and regeneration, to give us the many cell types that make up our tissues and organs. Those discoveries, the ongoing research, and the continuing advances have had a tremendous impact on human health, leading to many life-changing treatments. But for now, our journey must start at the beginning, with the essential formation of shape, known as morphogenesis. Hi, Helen.
Oh, hi, Courtney.
How are you doing?
I'm good. Welcome to the lab.
What are you working on today?
I can show you. Why don't you grab a chair? We can sit down and we can have a chat. These are just some adult fruit flies that we're preparing to set up for next.
So, Helen, I'm a PhD student also working on fruit flies, Drosophila. Can you tell me about this word morphogenesis?
Yes, so morphogenesis is a really fundamental process that happens in embryos and actually within tissues within the embryo that really helps them develop their shape as an embryo develops. Now, the fruit fly Drosophila has been really instrumental in helping us understand how these morphogenetic events happen. And that's because the fly embryo, just like a human embryo, undergoes many morphogenetic events such as dorsal closure and gastrulation, which really help us understand generally how different morphogenetic events in us happen. I have a model of an adult Drosophila here. And you can see, if we take this section of the body off, you can see inside the brain, the gut, the airways, the heart, many of the same tissues and organs that are within us. So dorsal closure in the fly involves the migration of two epithelial sheets over the surface of the embryo, and those two epithelial sheets move across the surface to seal a hole in the embryo, and they have to zipper up to close and seal that hole. And that's a very similar process to wound repair. And ultimately what we want to do is use the insights from our work in the fly to develop new therapeutic approaches to help us accelerate tissue repair in the clinic.
Hi, I'm Paul Martin. I'm president of the British Society for Developmental Biology.
Are you looking into the lens? Or...
No, I'm looking at you. I can't keep my eyes off you. You're so handsome.
Hi, Helen. It's nice to see you. What are you working on?
Oh, hi, Courtney. How are you doing? Sorry.
It tells us the differences between species. That's wrong. Let's go again. It was good, but she had the wrong hand and she knocked the chair. I knocked the chair. I walked into the chair. She ruined it. Courtney ruined it. Yeah.
It's getting less and less natural. Actually, we're gonna have Paul here telling us we must. I didn't say the word origami at all.
It is really beautiful, this science. It's better than physics.
That's definitely going to get a lot of people ringing more fields.
In essentially this, it's annoying, isn't it?
But I'd say, Pete, not again.
Did you get that?
It's a rat. Can't get rid of it now. Shankar, what have you done to my computer? You can see why I'm not actually featuring in the movie proper, just from those bloopers, right?
But it's not on the screen. Do you want to get something else?
Yeah, thank you. Income cool, right? So the highlight of the evening, though, the thing you all came for is the award of the Waddington Medal, which is the BSDB's premier prize. It goes to someone who is, you know, extra special. It's sort of like our Lifetime Achievement Award, but you've got to be super cool and nice as well as an outstanding scientist to get the Waddington Medal. Though how those two both got it, I beggars belief, but I wasn't the chair of the BSDB at the time. So this is the Waddington Medal. It was one that we've had for quite a long while, and there's this sort of silvery coloured one in the middle. Can you see that? Yeah. And tradition has it that we, you know, keep the person who's going to get the medal under wraps until the very last second. I'll tell you who the nominators are, Kate and James, and we thank them both. As I said yesterday, we need nominators, and we're always, you know, on the lookout for the next Waddington Medal winner. So please, if you think you know who should get it, then you know, you've got to tell Jens, nominate. And so I don't think you're going to guess who it is just from who the nominators are, and I suspect you might not guess from this picture of her as a young'un with mum and dad and a sister quite a while ago, I have to be said, in the Lake District where she was brought up, okay? And some of you might get it from this picture, but please don't clap yet. Some of you might get it from this, though I think she's somewhat in disguise. Her husband took this photo. They were in Turkey. He and she met as undergraduates at the University of Edinburgh. And he told me that they met in a genetics practical over a mouse, which most audiences would just think weird. But I suspect all of you will think how sweet and how romantic and how sensible to meet over a mouse. Okay. Now, sorry. I don't know, but maybe the recipient will tell you during her talk. It's a woman. I'll give that much away. She is an amazing exemplar of how to have a fantastic personal family life and a productive scientific career. It involves waking up, 3 in the morning or whatever. She has quite a few children, 4 children, and yet a very productive scientific life. Her sort of maternalness and kindness trickles down into her science. Everyone I speak to about her says she is, an amazing mentor and colleague. To typify her maternalness, several of us were at a scientific meeting that Phil organized in Singapore. And after that, we went for a brief holiday. Actually, I think the holiday might have been longer than the meeting, but we went to Thailand, and the winner is the only adult in this group, because she's looking after her 4 children, my 2 children, Phil's 3 children, and Jim Smith's eldest is there as well. So she's good at, being kind and looking after folk at home and at work. So when she'd finished her undergraduate degree in Edinburgh, she went off and did a PhD at ICRF with Gordon Peters. And she was looking at viral insertion sites in mouse mammary carcinoma cells or something that I didn't really understand. But she then went off to the States, to New York. She and her husband, who's also a developmental biologist, UK developmental biologist, went off to the States, and this is going to sort of give the game away, because she was hosted largely in the lab of Jane Dodd. And there she was very productive with several other scientists, and they pretty much nailed how the floor plate was induced and then how it went on to pattern the rest of the nervous system. The 2 huge papers in Nature and Science, which are sort of, they should feature in any of Jonathan's textbooks. They probably already do. She then came back to set up lab, as did her husband, who's Andy Furley. And I think they first moved to NIMR, and then Phil courted them off to Sheffield. And they've been there ever since. And the winner is, if you can read, Marisha Pleszczek. The winner is Marisha Pleszczek, and she has had several leadership roles in Sheffield. And her nominators flagged these 4 papers up as proof that she is still doing kick-ass developmental neurobiology. On her website, she says, I put my glasses on to read that her lab works on the brain, and on a particular bit of the brain, the hypothalamus, which is the master controller of physical, behavioral, and emotional homeostasis. So quite a big thing to be working on. It's a little bit of the brain. The winner of the Waddington is Marisha Pleszczek. Very nice for me to be able to award it to Marisha, because she's my mate, but she's also taking over running the BSDB at the spring meeting. We're going to hand over at the dance at the disco in the spring meeting. So the next few years will be settled and sensible after 3 or 4 or 5 years of turbulence. Not all my fault. There was COVID, you'll remember.
I plugged it in.
Oh,
No, it's there. We've got it.
Got it.
It's just a little bit slow.
Good.
Thank you, Paul, for that very kind, totally overkind introduction. This is a real honour. But one of the best things about this is that I get to talk to you for 40 minutes about all the wonderful people who I've encountered in the last 40 years. I can't believe you found that picture of me when I was 4 years old. I can't believe Andy sent you that. So, as Paul said, now let me just make sure I know how to work this. Where's the pointer? No, where's the pointer? There it is. So as Paul said, I started my scientific life, my real scientific life, as an undergraduate in Edinburgh University. So I went there to do molecular biology, which at the time, so this was back in about the 1970s, and you know, I don't look that old, but I am. So back in the day, molecular biology was the thing, and Ken and Nori Murray were absolute stars of the time. So I was, though, lucky enough to have as my tutor John Fincham, who was the professor of genetics. Now, in those days, can you believe it? We used to go and have tutorials at his house in his living room. But when he couldn't host us in his living room, he would host us in the most wonderful building on the King's Building sites, which was the old genetics library. This was a fantastic place. It was one of those quiet wooden libraries filled with books, and amongst the books were just books and books and books by Waddington, because of course he'd been the professor of genetics at Edinburgh before he moved back to Cambridge. So I was intrigued by these books. I just, there was something about them that was deeply inspiring, but I was certain that I was going to do molecular biology. So, as Paul said, from my undergraduate, I went to do a PhD at Imperial Cancer Research Fund, which at the time was at Lincoln's Inn Fields, with Gordon Peters. So this was now the early 1980s, and it should have been a really, really exciting time. Well, it was a really exciting time. Oncogenes, proto-oncogenes were just being discovered, and there was just this tremendous sense of excitement. And my supervisor, Gordon, was using a model. They were looking at proviral, they were looking at the way that mouse mammary tumor virus integrates at random. And if it integrates next to a proto-oncogene, then the strong promoter activates the transcription of the proto-oncogene, and it turns it into an oncogene, and the mice would develop with mammary tumors. So this should have been really, really, really exciting. And everybody around me was so excited. And they just talked about foci, foci, foci all the time. And you know, and we published papers showing that essentially the various genes that were called INT2 and FGF-HST were being activated and would promote tumorigenesis. But honestly, it just didn't speak to me. It just didn't. I mean, my southern blots were dreadful and my northern blots were even worse. And I know the whole thing. And it was not helped by the fact that at one point I broke my leg terribly. And my supervisor was not very happy about that, because in those days, if you broke your leg, it meant you spent a year in long leg plaster. Everybody in London knew me hobbling across Blackfriars Bridge with my leg in long leg plaster. Anyway, so I was saved, though, because our lab was shared with Jill McMahon. So on a Friday night, we used to go to the pub with Jill and her husband, Andy McMahon. And Gordon initiated a collaboration with Andy McMahon and David Wilkinson. David was one of the first people to be doing in situs in real embryos. It was so exciting. And suddenly I was given this, I was shown this picture of, look, this is the expression of INT2 in a real embryo. And it's in this thing called the neural tube. And that reminded me of something that I'd encountered as, you know, doing my undergraduate degree in Edinburgh, which was, of course, the Schweiman organizer experiment. So I was so excited. I thought, this is what is speaking to me. There's something about this that I just knew I loved. And again, I was lucky enough because soon after finishing my PhD, Andy and I got married. Now, you must note here my fine dissection skills, because this is going to come in again and again and again. Okay. So Andy's supervisor, Mel Greaves, was a very good friend of Martin Raff's, a fabulous neurobiologist. And so when I talked to Martin about just wanting to do something in neurobiology, he sent us to New York and introduced us to Tom Jessel and Jane Dodd. So we were, for those of you who know New York, we were way up on 168th Street, so way north of Harlem. It was, I mean, the first day I was absolutely petrified. The first day we arrived in New York was the only day in 6 years that I saw somebody beating somebody else against a car. And I just thought this was absolutely petrifying. And honestly, for the next 6 years, we didn't see anything like it. But I decided not to just turn heel and go straight back to England, because in Jane's office, there was the most fantastic poster of Madame Yvonne. It had this woman with this snake wrapped around her neck, and be original or die. And that absolutely typified and exemplified Tom and Jane. It's like, be original or die. They were absolutely original scientists, and the pair of them, absolutely fantastic scientists. So back then, you know, people weren't really making antibodies a lot, but Tom and Jane were amongst the first to do so. So they had been Tom had been trying to understand the basis for the sensory motor circuits in the spinal cord, and had decided that a good starting step would be to try to make monoclonal antibodies that recognized particular classes of nerves. And so this is one of the first antibodies that they raised, and it labels nerves or neurons and their axons, which are projecting from the dorsal aspect of the spinal cord down to the ventral aspect of the spinal cord. So Tom had a lot of ideas about what he wanted to do with this, but all of it necessitated somebody who could dissect. Now, what you don't know, and what Andy didn't know, is that when I was 10, my parents had sent me to a French convent school for 4 months to learn French, where every Saturday morning between 8 o'clock and 11 o'clock, we had to say samplers. And so I had really, really, really good dissection skills. So not only was I able to dissect embryos, which I immediately fell in love with. The first week I was there, I just thought these were the most exquisite things I'd ever seen. I just thought they were beautiful. And we dissected E11s and E12s and E13s. These were rats, I should hasten to say, not mice. But I just thought they just were so exquisite. And I still do. I must have dissected hundreds of thousands of embryos, and every time I dissect an embryo, I get excited about dissection. I just think they're so beautiful. But of all the most beautiful things in an embryo, the most beautiful thing of all is the neural tube. And in the neural tube, the most beautiful thing of all is the ventral midline. See, it is just this exquisite translucent structure which you can see, and there's just something about it which is gorgeous. So, given the pattern of growth of the axons, I mean, Tom was really clever. What he immediately realized is that these are developing, there's a bilateral symmetry to the neural tube. And he immediately conceptually thought, okay, if you're going to set up an organizing center to do that, you're going to set it up in the midline. And so he immediately started focusing on ventral midline floor plate cells and ventral midline roof plate cells as the source of organizing material. And in this case, his idea was that potentially the floor plate might make a chemoattractant that was guiding the growth of the axons of these dorsal neurons. He had, I mean, this was not the first idea that people were suggesting a chemotropic activity. Andrew Lumsden and Alan Davies had a paper not too far previously suggesting that neurons might be guided by chemoattractants in the peripheral nervous system. But this was the first time that somebody had thought to do that or to look at that in the central nervous system. So I sat down with Mark Tessier-Levine. So there's quite a funny story here. So Mark was actually in Tom's lab and I was actually in Jane's lab. But Mark he couldn't decide quite what he wanted to do. He was so excited about doing so much. So Tom had the very clever idea of getting me to start working on the same project as Mark, thinking that that would force Mark to take one direction or another. Anyway, it did, it worked. And Mark decided that what he really wanted to do was work on the chemoattractant. And it was brilliant. I mean, you know, collaboration in science is fantastic. And we had the best time. For a year or 2 years, we sat down every single day and we dissected. And I learned so much neurobiology from Mark because he was a neurobiologist and I was not. And he learned from me how to dissect floor plate. So it was a very, very happy combination. So what we did was to dissect out the dorsal neural tube and culture the dorsal neural tube as an explant at a time when neurons were just beginning to extend axons. And under these conditions, we didn't see any outgrowth. But when we cultured them with floor plate, which we also microdissected, we saw this beautiful outgrowth of the axons to the floor plate. And we were able to do fun experiments where we made the axons change direction by just changing the orientation of where the floor plate was relative to the normal direction of growth. So this led to these couple of papers that were the first to sort of show that axons were guided by long-range chemoattractants in the central nervous system. We then spent a year trying to purify this biochemically. So, you know, there are certainly times in science where you work for a long time and you get absolutely no results whatsoever. And this was the first of those occasions. A year we worked trying to isolate that biochemically. Absolutely nothing. The only thing we found, which did in the end prove useful, was that it had some ability to bind heparin. However, at that point, Mark left to set up his own lab. And as I'm sure you all know, he then in his lab dissected about 25,000 brains. And from that, isolated the chemoattractant and showed it to be the netrin. But I'd already decided that I was more interested in floor plate cells because, as I said, they were just so beautiful and you could see them. There was something tangible because they were translucent. And so at the time, I'd realized that although the floor plate in rat is beautiful, the floor plate in chick is even more beautiful. And they're slightly bigger as well, and it's slightly easier to isolate. So Catherine Tosney at the time had published these really beautiful scanning electron micrographs, which showed that the notochord lies directly beneath the ventral midline floor plate. Of course, the notochord Waddington had shown in chick derives from the organizer and had done some experiments to suggest that that organizing activity was maintained in the notochord. But this person, Van Straten and Hecking, Van Straten mainly, he had already done an experiment where he had taken a notochord from one embryo and grafted it adjacent to the lateral region of a neural tube in a second host. And he'd already obtained evidence that the notochord might induce a floor plate simply based on the fact that floor plate cells, as you no doubt are aware, have this very characteristic wedge-shaped appearance. And so based on the morphology of the spinal cord, he'd already published a paper suggesting that the notochord might have an impact on the appearance of a floor plate. Now, remember, at the time, we didn't have markers for floor plate. We didn't have any markers. But I thought, we do have a marker because we have a functional marker. We know that floor plate cells make a chemoattractant, so we have a functional property. And so I decided to do this slightly mad experiment where I grafted notochords next to a chick neural tube. You can see how that causes this wedge-shaped appearance of a cell type there. And then what I did was I dissected that little bit of tissue compared to a contralateral side, a control side, and I showed that the region next to the newly grafted notochord now possessed the chemotropic activity. So this was saying, well, functionally, there's a functional property there. And then one Christmas, I was back in the UK and I went to Claudio's lab for a couple of hours, I think it was, and he very kindly showed me how to dissect out, how to remove, do notochord removals, which is really pretty tricky. And I'm really grateful to Claudio for showing me how to do that. He showed me for a couple of hours, and then when I was back in New York, I spent 2 months where every day I just practiced and practiced and practiced, and they all just died. And then one day it all started working beautifully. And practice makes perfect when you're sewing or knitting or dissecting. So when we dissected up, when we removed the notochord and then tested the ability of the ventral midline overlying it, we found that then did not possess this chemotropic activity. So this was an absolute, we had a wonderful time in New York. I haven't named these people. None of them have got, apart from Mary, no, I've just said, apart from Mary, none of them have got any hair anymore, okay? But they were absolutely wonderful colleagues. And it's so nice to be able to keep in touch with all of them after all these years. So again, I mean, Columbia at the time, it wasn't just the labs, Tom and Jane's labs, that was such fun. There was a wider group of people. Liz was there, Claudio was there, Larissa was there, people in Frank Costantini's lab. It was just, there were so many people who were just doing such amazing science, and we had a lot of fun. But one of the unsung heroines of this time is this person here. So this is Susan Brenner Morton. Susan was Tom's research assistant, and she was a whiz at making antibodies. So many of the antibodies that I'm sure many of you have used over the years, the monoclonal antibodies, PACs, NKXs, they came from Susan. Susan made those antibodies. So she was the person who first made antibodies against floor plate and notochord cells. These are shown in rat embryos. And that then let me do an experiment, which was essentially well, I suppose these days you sort of call it an organoid experiment, but it's not really an organoid experiment because it's really using real tissue. So, sorry. So essentially the experiment was to take a piece of naive neural plate. And now I've offended a lot of people, which I didn't mean to. So the experiment was to take naive neural plate and culture it with explants of notochord or floor plate, and then see that in those recombinant explants, the notochord, illustrated here with an antibody against notochord, would induce floor plate cells. And we were able to show that the notochord induces the floor plate, and then the floor plate has homeogenetic inducing activity and is itself able to then induce some lateral cells to adopt a floor plate fate. But of course, Susan wasn't just making antibodies against the notochord and floor plate at the time. She was making antibodies against motor neurons and against neurons that are generally expressed in the dorsal spinal cord. And so using these antibodies and working very closely with Toshiya Yamada, who's shown here, we were able to do similar experiments, grafting experiments, removal experiments, in vivo experiments, and ex vivo experiments, all of which resulted in the idea that the notochord and the floor plate are the source of a long-range factors which can control dorsiventral pattern formation in the developing neural tube. So while I was busy doing my bits and pieces, of course, 10 years ago, 10 years prior to that, Jari Nusslein-Volhart and Erik Wieschaus done their amazing screen identifying the mutations that affect segment number and polarity in Drosophila embryo. Fast forward 7 years, and people like Roel Nusse were showing that these genes that played such an important role in pattern formation in Drosophila were playing really fundamental roles and were expressed in really interesting patterns in all sorts of places in the vertebrate. And I mean, I can still remember the day that paper of Roel Nusse's came out, because again, I mean, it seems extraordinary now, but back then, we just didn't think that genes that were expressed in Drosophila were going to have so many homologs in vertebrates. I mean, it seems impossible now, but it's true. I'm not making that up, am I? I can remember Rob Crumlaff showing the Hox genes, and it was just an extraordinary time as people realized just this massive conservation. And then, of course, you had people like Phil sitting in the front row there, who was doing all sorts of really beautiful experiments that was showing how these genes were affecting the patterning of the Drosophila embryo. And so I understand that at a meeting, can't remember what you said it was now, Phil, but it was some meeting where Phil with Andy McMahon and Cliff Tabin came up with the idea that a Drosophila gene called hedgehog might have a vertebrate counterpart. They identified sonic hedgehog and found that it was expressed in the notochord and the floor plate, and did some experiments that suggested that it was the factor that was mediating the ability of the notochord and floor plate to pattern the dorsiventral axis. So note the timing. These papers came out in December 1993, and shortly after, from Tom's lab, Henk Rowling was the first author on a paper that again showed similarly that floor plate and motor neuron induction is mediated by this same gene, which now we all know as sonic hedgehog. I still don't like that name, but as these really lovely images show, sonic hedgehog is first expressed in, at least at posterior levels, is expressed in the notochord, and then a short time later in the notochord and the floor plate. And I think one of my main contributions again in this paper was to show that if we took COS cells transfected with sonic hedgehog and cultured them next to naive neural plate, then we were able to induce the differentiation of floor plate and motor neurons. So that was really exciting. I mean, that was just, it was astonishing again to see the transition from phenomenology to the sort of a molecular understanding of pattern formation. And of course, soon after, so I left the lab around about this time. I was leaving around about this time, and shortly after, James Briscoe joined the lab, and I don't have to tell this audience what an amazing job he's done over the last 20 years of really illustrating how that braided sonic hedgehog signaling results in the specification and the patterning of cells along the dorsiventral axis of the neural tube. It really has become a paradigm for our understanding of morphogen activities and morphogen signaling over space and over time. But what I really want to turn to now is this paper that came out some years later. It was published in 1996 from Phil Beach's lab. And this was when sonic hedgehog was first knocked out in the mouse embryo. And the phenotype for me was really interesting because one of the main phenotypes that was obvious was this holoprosencephaly and cyclopic phenotype. In other words, the sort of a major phenotype that was obvious was a forebrain defect. So the reason that I was so interested in that was because, so forgive me for showing another picture of Tom, but for those of you who knew Tom, you will, if you ever went into his office, you would remember seeing that his office was covered in whiteboards. And against the background of those whiteboards were always just covered in little illustrations and scribblings of what could be going on. It was like his thought processes were on this whiteboard. And that sort of filtered through into everybody in the labs, in Tom's lab and in Jane's lab. So if I look back at my lab books from that time, they're absolutely full of little diagrams of embryos and what was I seeing. And so you can see this one from 1991 and this one from 1992. This was a picture, this was my, I wasn't very good at doodling, not like Rosa. I mean, but this was my picture of a chick embryo, and this was my picture of a rat. And you can't read this, I don't think, but what it says is, midline, it says midline goes all the way up, but very difficult to know if this is floor plate or notochord and where it ends. And so I just was really fascinated by the fact that this midline cell group went all the way up. And as I say, you could see it because it's really obvious because it's translucent. And at the time, of course, I mean, experiments, there'd been quite a lot of debate going way, way back, you know, hundreds of years as to where the floor plate officially ends. But pretty much the consensus was that the floor plate proper ends in the midbrain and that there is not a floor plate-like cell extending into the forebrain. So I thought, well, that there might not be a floor plate, but there's certainly a midline group that looks pretty much like floor plate. And so at the time, some really lovely work coming out of the Pueyes and Rubenstein's labs were basically showing, you know, again, uncovering lots of different genes now and showing that the early neural tube was patterned along its anterior, posterior, or rostrocaudal axis into defined regional domains. And so I'd done some experiments with largely with people in Thomas Edland's lab, really, where I'd shown them how to dissect pieces of naive neural plate from different rostrocaudal domains along the anterior, different domains along the anterior posterior axis of the neural plate, and then exposed them to sonic hedgehog. And that had shown that there is an element of pre-pattern and that sonic hedgehog can induce the differentiation of particular ventral cell types that depends in part on a manner in where they lie along that anterior posterior axis. But somehow there just seemed to be something that was missing. I just couldn't believe that sonic hedgehog was the same all the way up. The neural plate, the midline just didn't look the same all the way along the axis. So this was all happening around the time that I moved to my first independent position at the NIMR. It was lovely. We were way up in the attic here. It was a beautiful little lab. It was tiny and it was lovely. And I could look out and pretend that I was on a mountain. This was quite a busy time because by then we'd had David and then we had David and Nikki, and it would have all been a disaster in the lab if I hadn't had two absolutely wonderful people, Nusrat Sattar, a research technician, and Kim, my very first PhD student. This was taken at Nusrat's wedding. We all, Kim accepted, the rest of us just looked like sparrows next to these absolutely exotic, you know, wonderfully colourful Nusrat. So at the time, there'd been a really lovely paper from Roswitha Seifert showing that in the chick, the notochord at its anterior limit becomes the precaudal mesoderm and then precaudal mesendoderm. And then Claudio and his colleagues had found that in the chick, as in other species such as Xenopus, the precaudal mesoderm could be recognized through its expression of goosegoid. So, as I say, one of the things that we could do really well in the lab was to dissect. And so we worked out a way of dissecting out first mesendoderm and then uniquely from that, dissecting out uniquely the mesoderm. So we would dissect out this rod of axial mesoderm composed of notochord and then precaudal mesoderm, which could be removed from the underlying endoderm through an enzymatic treatment. And once you'd removed it, it sort of sprung into this lovely little Y-shaped shape. And if we cultured that, for instance, we could see just the different behaviors of the notochord and the precaudal mesoderm. The notochord undergoes convergent extension, whereas the precaudal mesoderm is mesenchymal-like and exploratory. So very fundamentally different cell behaviors in these two cell groups. with Kim and Nusrat, we did an experiment where we asked whether precaudal mesoderm could induce specifically the characteristics of ventral midline floor plate cells that develop in the forebrain. And of course, by now people were beginning to identify markers that would distinguish ventral midline cells in the forebrain and ventral midline cells in the posterior neuraxis. So floor plate cells proper compared to forebrain ventral midline cells. And we were able to show in a variety of ways that the precaudal mesoderm was able to induce specifically ventral midline cells of forebrain character. Shortly after, Kate, working with Claudio and Pera and Kessel, had some really lovely studies showing similar, coming to a similar conclusion through in vivo experiments. So with Kim, one of the things we did was to figure out that the ability of the precaudal mesoderm to direct the differentiation of forebrain ventral midline cells was due to the expression of sonic hedgehog cooperating with BMP7 from the precaudal mesoderm. So in contrast to the notochord, which was exclusively expressing sonic, the precaudal mesoderm co-expressed sonic and BMP7, and those were working together to give forebrain type ventral midline identities. So I want to mention this. So again, we had great colleagues at Mill Hill, but one really special colleague was Larissa Pevney. Larissa we'd known her as a PhD student when she was at Columbia. She then came to work at Mill Hill, where she was with Robin Lovell Badge. And Larissa, in Robin's lab, they had just identified, with Peter Goodfellow, they'd identified the SRY genes, but it was Larissa who really showed that SOX1 is a marker of early neural tissue. And so she came to my lab to do some of those experiments and to really show that SOX1 was expressed in neural tissue, and then devised a set of experiments that would show a key role for SOX1 in neural determination. So yeah, Larissa was just such a wonderful, wonderful colleague. And yeah. So Mill Hill was great. And there were two things though. London is flat and I couldn't teach. I just love teaching developmental biology. So when Phil asked whether we were interested in joining the developmental genetics program, we were absolutely delighted absolutely thrilled to join him. So Phil, with his typical charisma and energy, had managed to recruit all sorts of really wonderful colleagues to Sheffield, working on all sorts of model systems. Many people working on mouse, Drosophila, zebrafish. I was the only chick lab, but hey, you know, it didn't matter. They were just such great colleagues, and there were hills to climb. So that was just an added bonus. And there were students to teach, which was even also great. So at the time, Kim came up and was with me for the first few months in Sheffield, but then Ian Patton, a student, joined when Kim left. And he took over from Kim. And it was really through his work, which I'm just sort of summarizing here, that we really began to appreciate that these ventral midline floor plate cells that form in that anterior part of the axis are actually going to be part of the hypothalamus. So that it was really Ian who first we really appreciated that through his work, and also began to appreciate the different nature of hypothalamic floor plate cells in comparison to more posterior floor plate cells. So they express sonic, but they express a whole array of other markers, including different transcription factors like NKX 2.1. So here you can see its expression. This is the precaudal mesoderm. You can see it's just wide, it's bigger. There's the hypothalamic floor plate, and they also express a number of signaling factors that are exclusive to them, such as FGF10 and BMPs. And Ian was able to show through some really tricky experiments where he ablated precaudal mesoderm that if you ablate precaudal mesoderm, you don't get rid of the neural tube entirely. You still have neural tissue, you still have telencephalic tissue, but essentially what you don't get is the induction of the hypothalamic floor plate cells. I found this ancient picture, which we never published actually, which was a precaudal mesoderm ablation that we did maybe 20 years ago. But the reason I'm just showing it here is because one of the things that you don't really get from this sort of image is that when you ablate precaudal mesoderm, it's not that you just lose expression of hypothalamic floor plate markers, but you really change the morphology of the brain. Essentially, you just don't get proper eye morphogenesis. So we'll come back to that later. So then there was a period of years which were frustrating, actually. We had, again, wonderful people in the lab. Pam Ellis, a research assistant, Kyoji Oyama, who's just the most wonderful postdoc. And essentially what they were trying to do was really work out how the expression of this complex array of signals, so sonic, BMPs, FGFs, might be patterning the hypothalamus. So I really wanted to work on the hypothalamus. I just thought it was a very poorly understood region of the developing central nervous system. It's sitting in such close proximity to the periphery. So in development, the hypothalamus sits adjacent to Rathke's pouch, which is going to give rise to the anterior pituitary. And in the adult, the hypothalamus, again, you can see it's sitting here in the very, very base of the forebrain next to the anterior pituitary. And of course, I don't have to tell you what the important roles of the hypothalamus are in survival of the individual and survival of the species. It's mediating aspects of homeostasis that people used to think was just physical homeostasis, but as Paul said, it's now understood through work in mouse that a lot of mental aggression states, behavioral states are also mediated by hypothalamic neurons. So understanding the development of this region is really important, and nobody looks at it. So we were trying to work out how these factors might impact on hypothalamic patterning, and I think we were just stuck with the idea that it was all going to be pattern. That was a mistake. I think we were just thinking that it was all about pattern progenitors, and then they all expand isotropically. And so the other thing that became very obvious was that along the anterior posterior axis, the pattern of sonic is really quite complex. It's expressed in a broad ventral midline in this region, not expressed here, then re-expressed there. And so we made some progress, but it was frustratingly slow. And actually, probably that slowness was also due to the fact that by then we'd had two more. We had Nikki, David, and now John and Caitlin. And so luckily, again, I was rescued from this state of completely going mad by two other fantastic PhD students, Liz Manning and Raman Das. And so Liz just has amazing hands in the lab. I mean, she just amazing hands. I don't know whether you did embroidery when you were little, Liz, but anyway. And so she immediately just did some beautiful fate mapping experiments targeting the forebrain, the hypothalamic floor plate cells, the cells that we're coming to already think were hypothalamic floor plate cells, and then tracing those, letting those embryos develop, and showing that indeed that these cells, this really is the origin of the hypothalamic floor. And then she, working with Kyoji, went on to show that at least one facet of the role of BMPs is to induce the expression of transcriptional repressor TBX2, which then Doug Epstein in mouse shows directly represses sonic hedgehog by displacing a SOX2 from the sonic hedgehog promoter. And so that at least gave us some insight into how you get this little patch of sonic hedgehog negativity. And at the same time, through her work, Liz began to show the first idea that actually, as this happens and sonic hedgehog is downregulated from ventral midline cells here, they suddenly begin to undergo this extensive proliferation. So at the time, there was quite a lot going on. So Phil had very successfully transformed the developmental genetics program into the MRC Centre for Developmental and Biomedical Genetics. And really what this meant was engaging with the local clinicians to teach them all about how wonderful developmental biology was and how useful especially zebrafish and fly models were for thinking about how to generate innovative disease models. So somewhere along this period, Phil went to Singapore for a year, and he did come back sort of on and off. Thank you, Phil. But there was quite a lot to do in the meantime, and I sort of stepped in to generally, keep things going as the MRC Centre went through to its end and then evolved into the Bateson Centre, which then I was very glad to see Steve Renshaw taking over in 2015. So it was great because actually we had a very thriving community of developmental biologists working closely with clinicians. That community still persists today. It's a wonderful atmosphere and just a lot of people doing wonderfully exciting stuff. So, you heard this morning about, so we've got Emily Noel working on heart, we've got Kira Campbell, we've got Matt Towers, new people coming in all the time and doing great science. So I was a little bit schizophrenic at the time because I was sort of thinking very hard about Drosophila work and zebrafish work on top of the chick work. But I think there were three things that I really gained through that experience. One is all of the wonderful outreach that we could do. And I just think the film is just fantastic because, getting across the idea that developmental biology is so important to the wider community is something that we all have to work so hard at. But the models that we have and all of the talks today and the beautiful data and those beautiful just the videos, I just think that, that we've got to get those out to people. It's such a beautiful system. And I think Waddington would be delighted to see the way it's going, because one of the books that was in the genetics library is this book here, where he was just talking about the importance of the natural sciences and art going hand in hand. So it was great. It was great to be leading on some of the outreach events that we could coordinate over this period. The second thing that it did was give me a lot of contacts in the zebrafish field. And then the third thing it did was really, through the clinicians, make me start to think hard about the importance of the cells, the bits of tissue we were looking at for later function. And so one of the things that we began to think is, well, what are these cells that are downregulating sonic going to give rise to? And as well as downregulating, at the same time that BMPs cause the downregulation of sonic, they cause the upregulation of FGF10. And we were able to follow FGF10 cells by lineage tracing over time and show that they give rise to this wonderful little structure here called the infundibulum. So the infundibulum is a sort of a ventral outpocketing from part of the floor of the hypothalamus, and it's going to give rise in the adult to this region here, which forms the interface of the brain and the body. It's probably the primary interface of the brain and the body. And that interface occurs when axons from hypothalamic neurons grow towards the infundibulum and portal capillaries grow towards the infundibulum to meet, and then the axon, the end feet of the axon terminals release neurohormones and neurotransmitters, which are then picked up by the capillaries. So the coming together of the axon end feet and the capillaries is essentially what is creating that brain-body interface. And so there'd been a paper a few years before that, showing that FGFs can guide motor neurons. And so we had the idea that potentially FGFs might be guiding the neurosecretory neurons of hypothalamic neurons. And in a series of a collaboration with Matthias Hammerschmidt, we combined studies in chick with studies of his really beautiful zebrafish lines to show indeed that, and I'll just sort of make the point on the zebrafish slide. So you're looking at sort of a ventral view onto the surface of what would be the fish equivalent of the infundibulum. And here is this little network of capillaries, and in green are the end feet of the axons. And in an FGF3 mutant, then neither the capillaries nor the end feet come. And so that was quite pleasing, because it really suggested how a simple molecule is integrating the construction of this interface, which is so important for brain-body and body-brain communication. So Caroline Pearson, at the same time, another PhD student, was interested in the idea that FGFs might be regulating proliferation in those cells. And that was partly based on Liz's finding that we saw this upregulation of proliferative characteristics as the cells downregulate sonic hedgehog. And at the time, people like Fred Gage and Fiona Dutch were identifying, were performing this thing called a neurospherogenic assay, where essentially you could take cells and dissociate them, plate them at very low densities. We didn't go as low as clonal densities, but we went pretty low, incubate them and ask whether they give rise to neurospheres, which then self-renew and self-renew and self-renew. And so Caroline was able to show in quite old chick embryos that FGF10, this FGF10 positive domain, was the source of cells that would form neurospheres, which would self-renew. And very nicely, in contrast to neurospheres that we were able to derive from other parts of the nervous system, the subventricular zone or the spinal cord, they expressed particular characteristics that said that they were hypothalamic in identity. So, as I say, at the time, people like Fred Gage, there'd been this idea that, there were no stem cells in the adult brain, but people like Fred Gage, Verdon Taylor were identifying stem cell niches in the subventricular zone and the dentate gyrus. So I had this mad idea to see whether FGF10 positive cells might be retained in the adult mouse, and that they might be stem-like cells in the adult mouse hypothalamus. So I wrote a BBSRC studentship grant, actually, to test that idea. And in fact, 2 PhD students joined my lab at the time. Sarah Robbins was funded through the BBSRC studentship, Ian funded separately, and they both took this seriously and they ran with it. I mean, they absolutely ran with it, and they were able to now. They didn't do this in the adult chick because we don't keep adult hens at Sheffield. So they switched systems to work on the adult mouse, and they were able to show indeed that if you sort of follow this region, it's the top bit of the infundibulum here that's going to give rise to the median eminence in the adult, that FGF10 is retained in the adult, where it's expressed on these beautiful radial glial-like cells called tanocytes. We were able to show that tanocytes continue to respond to FGF signaling by enhanced proliferation, and through conditional tracing, we were able to show that under certain conditions, tanocytes will give rise to new cells, including astrocytes and at very low levels, neurons, which then migrate along the tanocyte processes to contribute to cells in this nucleus here is the arcuate nucleus, and those neurons are involved in energy homeostasis. So quite a lot of labs now have taken this, and quite a lot of the clinicians are quite interested in this idea that there might be a hypothalamic niche which is diet responsive. I'm still very much not certain whether I think there is a hypothalamic niche that is important. I think it might be important in some species. There's been some really nice studies in sheep showing that, which have a very seasonal reproductive cycle, showing that there are new neurons born at particular reproductive times. So I think in some animals, there is good evidence that there is a stem cell niche in that region. Humans have those cells there, but it's not clear whether they are actually operating to mediate what we'd call allostasis, which is the ability to change over life. But I think it's an interesting idea. However, I'm a developmental biologist, and I always wanted to really know what was happening in the embryo. And so really what this led to were studies that we did with Matt Towers and a PhD student, Travis. So we recruited Matt from Cheryl Tickle's lab, and he was really fascinated by how FGFs might act to mediate timing in the limb, actually. But we began to ask whether, really, we were interested in whether the FGF10 cells that we'd identified might act as in some type of stem-like manner. And through experiments that Travis did, which involved a combination of fate mapping and interference experiments, Travis was able to show indeed that FGF10 cells can to some extent give rise to more FGF10 cells, but at the same time are also capable of giving rise to daughters that will then migrate anteriorly and become the sonic hedgehog positive cells that we'd identified in these anterior regions of the tuberal hypothalamus. So what this essentially said has been covered in detail in the talk that Kavitha gave. So I'm not going to go through that in detail again, but simply to say that really our whole understanding now of how the tuberal hypothalamus arises has evolved from being one where we think that the whole thing is being patterned to one where we now understand that there are elements of patterning, but also elements of proliferation and the directed movement or migration of cells against a wave of, so a wave of differentiation against a wave of signals that maintain some sort of stem-like niche. So round about this time, round about 2018, I went to a meeting, and it was a meeting where sort of the only other person in the world who's really interested in hypothalamic development was, and that is Seth Blackshaw. So his lab works and has worked for many years trying to understand hypothalamic development in the mouse. And way back in about 2018 at this meeting, Seth told me that they'd done a single cell RNA sequencing experiment that identified really some of the key progenitor domains that will give rise to different areas of the hypothalamus. But he was very clear about the fact that although this paper said we've covered the whole of hypothalamic development, he was very clear that they actually had not covered the very early stages of hypothalamic development. And so we collaborated to dissect out the hypothalamus. So we isolated neural tissue and then dissected out hypothalamus at 6 different time points from the developing chick embryo, and did a very large scale single cell RNA sequencing study. So this was in collaboration with Seth and his postdoc Dongwon or Thomas Kim, who's now set up his own lab at Aarhus. And in Sheffield, Sarah Burbridge, Elsie Place, and Ian Groves came to help to do this. And I was really delighted when, having been away for many years doing other postdocs, Kavitha and Liz returned to the lab to help us with this absolutely mammoth project. And it was a mammoth project. It took us, so there were about 3 of us dissecting hypothalamus 3 days a week for almost a year to generate the tissue that we needed. So what did we get from this? Well, we just got really great single cell RNA sequencing data, but it would have been the thing that really made the difference, of course, was this fantastic HCR that just came about the same time, and which, you know, Elsie really pushed in the lab. So this just let us, for the first time, really analyze the co-expression of multiple genes at once. And for those of you who haven't done this, I mean, it is extraordinary because you can analyze 3-4 genes at a time, and then you can strip the sample and analyze again and again and again. And, you know, often we're looking at 12, 14 genes on exactly the same sample. And one of the things that I really want to point out is that you probably can get an idea from this of just how difficult it is to image the hypothalamus, because it is so curvy. It just is, it's just really difficult to analyze it from any single view. So again, we did a huge amount of work to validate the expression of every gene in numerous different views. So transverse sections, sagittal sections, whole mount views, hemi views, and oftentimes we would isolate neural tubes and then do the HCRs on the isolated neural tubes to just really generate a complete picture that essentially let us then develop these rather you know, well, you have to in the end put them onto simple 2-dimensional cartoons, but at least we began to get an idea of how the hypothalamus changes over time. And one of the really nice things about this was that we were able to compare our very latest time point with the earliest time points that had been analysed by other labs in mouse and human. And this just showed an incredible conservation in hypothalamic gene expression. So everybody always thinks that the hypothalamus is one of the most evolutionary ancient parts of the brain because it's been conserved, because it's so important in regulating your life. And indeed, I think this really provides very good evidence for a lot of conservation at neurogenic periods. And so subsequently, we've dissected early stages of mouse now with the Blackshaw lab. So hopefully we'll find out very shortly whether that conservation is true at the younger stages as well. So I just want to end with a couple of, you know, the key things, because I mean, the problem with this single cell RNA sequencing stuff is that, like, you're in a child with a sweet shop. They've got so much you could look at. So what do you decide to look at? And I think for me, there were two, I'm just going to point out really the outstanding thing that emerged from this, because at our very earliest time point, essentially the clusters fell into 2 key categories: hypothalamic-like cells, shown in red here, and then in blue, cells that had the characteristics of diencephalic prethalamic-like cells. And RNA velocity indicated quite a strong trajectory from prethalamic-like to hypothalamic cells. And one of the genes that we identified as being expressed in the prethalamic cluster was a gene that nobody had remarked on as being expressed in that part of the neural tube before, and it was follistatin. And so you can see this is an isolated neural plate from an early embryo. We're looking at follistatin and then sonic and NKX 2.1 marking the hypothalamic floor plate cells. And you can see how they are essentially eliminating the expression of follistatin from the midline. And we did a bunch of experiments that suggested indeed that follistatin will inhibit the induction of hypothalamic floor plate cells and must be inhibited. And I think that this is probably another key role for BMP, although I should say we haven't actually proven that yet. But for me, I think it was just great. It was like doing this massive jigsaw puzzle over 30 years, because suddenly I could say, yeah, now I know what the end of that, I know what that little structure is, that is the developing hypothalamus coming. So it was just great. But more importantly, going to, what do you do with all of this data? So all of our data was suggesting that hypothalamic cells are coming from a relatively posterior part, from a region of the neural plate, neural tube, that at later times will lie relatively posterior. And so, you can't really rely on RNA velocity. You've got to do the proper fate mapping. And so in just the most phenomenal sets of experiments, Liz with Elsie over the last couple of years have performed an extensive fate mapping analysis of the ventral neural tube. And surprisingly, this just hasn't been done before. So a lot of fate mapping has been done, but primarily of the dorsal aspect of the neural plate and neural tube, not the dorsal aspect of the neural tube, not the ventral aspect. So now with our array of new markers, I'm just showing a couple of examples here where Liz can really target with exquisite precision, based on everything that we know now about the morphology, follow those cells. So in this case, this is where the eye cell goes, and then look at the fate of cells in the context of an array of hypothalamic markers. And essentially what Elsie has then done is taken those images and made them into a series of essentially model. They're not really models, because they're all coming from the real data sets that are really giving us a really good idea of how the forebrain undergoes its morphogenesis. And I think the single key point that I want to make is that the pattern of growth in the hypothalamus is completely distinct from the pattern of growth in the telencephalon in dorsal aspects. So in the hypothalamus, as you can probably see from, say, this image here, or this image here, everything, and then from these lines, everything is, the growth is all in this direction. It's all anterior posterior, whereas in the telencephalon, it's dorsoventral. Okay, so it's completely different. So what Elsie has done from this is essentially put these together in a way where we can now really begin to look at the morphogenesis of the brain completely. And I think, again, a take-home message would be that the growth of the hypothalamus is absolutely intrinsically linked to the morphogenesis of the eyes. The one follows the other. And I think that's why I wanted to show you that example early on, showing, the precaudal mesoderm, you ablate it. It's not just that you don't get fate, but you don't get the morphogenesis of the eyes. So the reason that we think that this is important is because it completely contradicts the model of forebrain development that has been held for the last 30 years, because the current model of forebrain development suggests that essentially from the neural tube, you get essentially differential proliferation, so heightened proliferation in dorsal region, transforming these blocks to these wedge-shaped cells, and essentially a posterior rotation. And in this model, the hypothalamus, which is in pink here, is situated beneath the telencephalon and is separate from the prethalamus. Whereas all of our data, so first of all, the single-cell RNA sequencing data, and then more recently, the fate mapping, which I think, is just you can't argue with fate mapping experiments. They show exactly the opposite, which is that the hypothalamus is, as its name suggests, situated below the thalamus and the prethalamus, and that the rotation is absolutely contrary to what is currently accepted. And I think the reason that it's all got muddled up, as I say, is because people have done fate mapping in the dorsal part of the spinal cord, but just nobody's done the fate mapping in the ventral to understand what's going on. So again, if you want to see those, go and have a look at the posters. So really, I just think, so I often say to my children, I just think I'm the luckiest person in the world, really. I can't imagine. So the mouse that we met over was a Danforth's short tail, and I was too scared to pick it up and check the plugs. And Andy was brave enough to pick up the mice. And so, you know, I just can't believe how I've gone on this fantastic journey from a Danforth's short tail mouse to just being able to explore the absolutely fantastic ventral midline and hypothalamus, and how at the same time I've managed to do that within the context of just this amazing family. I've just been so incredibly lucky. And of course, that's partly been because I've been funded, and I'm just, you know, hope that we can go on getting developmental biology funded. And it's been because over the years, I've had fantastic people in the lab. I haven't been able to introduce all of them and all of their work, but thanks to all of them. And also, I think the undergraduates sort of keep me young and happy as well. And one of the things that I think is particularly nice is to see undergraduates who've done summer projects in the lab and then have gone on to do PhDs and start running their own labs. That's just been a real pleasure. So thank you very much indeed.
Good. What a wonderfully inspiring journey. Totally worthy winner. Think about who you think should get a medal in the future. Quite a tough act to follow. I think the bar is open. I don't drink, so I don't really care, but is the bar open, Sally? You do drink. Yes. Congratulations, Marisha. Fantastic talk.